Morning, good after evening, something like that.
In progress.
Hello, happy Valentine's Day, everybody.
Happy Valentine's Day.
Happy Valentine's Day.
Happy Valentine's Day.
Happy Valentine's Day, y'all.
Happy Valentine's Day.
So first, if you didn't see my email last night, it was brought to my attention that even though I posted on the syllabus that a lab quest would open last week, I didn't actually open it. And so it's now there.
Yeah, I sent you a message.
When did you send it to me?
Let me see. On
Was it
Thursday.
last night?
No, it wasn't Thursday because I was reading the syllabus and it says that it was going to be open on, I think.
It was supposed to open on Wednesday.
Yeah.
All right, I didn't get the message, and I can go back and look, but I didn't get it, and I know that sometimes I just don't get them.
Okay.
I'm recognizing that.
I just was asking about the lab.
Okay.
Number 2.
Well, it's there, and it isn't going to close this week because I didn't open it until last night. Therefore, it's not going to close. I don't want to just give you a couple days to do it. I don't think that's fair, especially if I didn't open it when I said I would.
Yeah, I read that it was going to be closed tomorrow. So.
And that isn't the case. I'm going to leave it open another week.
Okay. Okay. Thank you.
And so it's just covering the 2 lab exercise 6. And tomorrow night we'll go through the integumentary system. Questions, concerns? I can tell you today computers have been the bane of my existence for the last like 24 hours. And so we're going to see how tonight goes over. I hope it goes well. Or I think I'm just going to shoot myself before the end of the night. Don't say anything though. You know that feeling, right? I had to do a project last night and it wasn't something that my monitor could do. And so my dad actually gave me a new monitor in November, but I haven't put it up because that's the way I am. It's just kind of been taking up space on the floor until I set it up. And then once I set it up, I realized it wouldn't do what I needed it to do anyway. And now it's not doing what I needed to do again. And I know it's that he has it programmed a specific way and he's the master of computers, but I don't know how to disable whatever he did. So computers are not my friend right now. So let's see what happens here. Sorry, just venting for a minute. I've been venting for like the last half hour to my roommate here. Who just tolerates me. Let's see. So we will finish up chapter 2 tonight. And we will most likely start chapter 3. Let's see what happens. So inorganic versus organic compounds. Inorganic compounds are simple structures, small size, small molecular weights with few atoms involved. And there's always ionic or covalent bonds involved. Some examples are water, sodium chloride, and carbon dioxide. I don't know what I was thinking. Organic compounds are what we mainly focus on, even though we know that sodium chloride, water, and carbon dioxide all have very important physiological roles. Organic compounds are what, when you study chemistry, you can spend a full year talking about organic chemistry and how it differs from inorganic chemistry. And the reason, what we like to think is that organic chemistry is typically the chemistry of biology. That isn't completely correct, and that's a bad generalization when you consider that sodium and chloride and the carbon dioxide are all key to physiology and its function. So inorganic simple structures, ionic or covalent bonds, organic compounds, carbon-based, and they must have bound hydrogen within them. It can be chains of carbon atoms and also contain nitrogen, sulfur, phosphate, and oxygen. Typically has a large size, complex structures, and only covalent bonds. So a major distinction between inorganic and organic compounds is that organic compounds are only covalent bonds. And in organic chemistry, organic chemistry is capable of synthesizing macromolecules and polymers. One thing that I want you to really get out of this, and just a very simple view, if the compound doesn't have both carbon and hydrogen, it's not an organic compound. It has to have those two elements. If it only has carbon or it only has hydrogen, it's not an organic compound. If it's got carbon in it, it's got hydrogen in it, and it's got a bunch of other things, it's an organic compound, but it has to have both carbon and hydrogen. In my biology course my general biology course that I teach, one of the first things we talk about is the fact that, and it's the best exam question, because you give them a series of different structures and only one of them contains carbon and hydrogen, and say which one of these is organic. And people will think, well, if it's carbon-based, it's organic. And that's not the case, because you look at carbon dioxide and it's not. And that's not an organic compound. So just remember, always contains carbon and hydrogen. So why carbon? Why is that the basis of organic chemistry? Why don't we use potassium? Why don't we use zinc or chloride or sodium or oxygen? Why do we use carbon? Before I show you this, I want to get something. Pull up a periodic table, and you may not be able to see it really well, but the example will be there. We use carbon as the basis of all life. All physiological compounds are carbon-based. Big macromolecules are carbon-based. If you look here, and I know this isn't very big, but I have a periodic table here. And you remember that these 6 elements right here that are, as I scan on them, are becoming bigger. These are the electronegative elements, right? These are the 6 over here that we worry about. Carbon is right next to them. So carbon theoretically could be an electronegative atom, but it's not. It's not. It is electroneutral. When we think about, remember what electronegativity means. What does electronegativity mean? Someone tell me, what does it mean for something to be electronegative? So if it's electronegative, it means that it, in a covalent bond, it will work to attract the electrons more towards its nucleus than the other atom that's involved in the bond. It's going to have a stronger pull on those electrons. Carbon doesn't do that. It doesn't pull on them. It doesn't give them up, so it won't form ionic bonds. It only will share them, and it only equally shares. It also has 4 valence electrons that bind with other atoms so that it can be provided with up to 4 more electrons to fill its valence shell. So it can make up to 4 other double bonds. So it's electroneutral, meaning it only shares its electrons. And remember that the covalent bond is the strongest of the bonds we talked about. So it only is capable of making covalent bonds. And it can make up to 4 of these, which allows it to be very versatile and allows it to make multiple different shapes. So let me ask a question. What elements must an organic compound contain?
Carbon and hydrogen.
Correct. What types of bonds does carbon form with other atoms?
Covalent?
Covalent, right. How many bonds is it capable of making?
4 more?
More. And what types of structures can be made with it? Anything that involves covalent bonds. trying to think if there's, as long as that molecule is capable of making covalent bonds, it can bond with carbon. Trying to think if there's any examples where it can't, and I can't think of anything. So the fact that it can make these 4 bonds, and if you think about that, that allows it to work, essentially work in any dimension. Carbon skeletons can come in a large variety. Carbon skeletons meaning only carbon and hydrogen are present. So here's the carbons, here's the hydrogens. When there's only carbons and only hydrogens present, it's a carbon skeleton. Carbon skeletons can vary in length. They can vary from 2 to 3 to 12 to whatever. We can also get double bonds, or even, trying to think, we can get triple bonds too. We can get, so we can get covalent single bonds, double bonds, and triple bonds, which can vary in location of where those double bonds are. So these are isomers of one another. They have that same chemical formula, but different structures. Butane and isobutane, 2 other isomers. So just based on their branching pattern, same chemical formula, and they can be branched or unbranched. We can have rings. We can have rings that have double bonds in them, or rings that don't have double bonds in them. But the one thing that's important here, outside of the fact that carbon is great because it can make all sorts of shapes, allows you for them all the amazing variety that we have in biology of shapes and structures and molecules. But it's boring. And the fact that it's electroneutral, when it, if you remember atoms that have a 0 charge on them, where the number of protons and the number of electrons equals and their valence shell is filled, they don't react with anything. You know, I mean, unless you can have a good explosion, why do you even have it around? Right? And so it's being, the fact that it's electroneutral means that it doesn't do a whole lot of things. It provides a lot of different structures, but it's not going to react with anything. So carbon skeletons are great, except they don't really react with things. And to have biology and have life, we have to have reactions. Therefore, we add functional groups to these carbon skeletons. So we add little small clusters of atoms, many of which contain electronegative atoms, to give organic properties, to give organic molecules different chemical properties and allow them to be reactive. Do you remember that what's one thing that's found in all polar bonds? First off, what is a polar covalent bond? What has to be present for it to be a polar covalent bond versus a non-polar covalent bond? Somebody tell me, what's the difference between a polar bond versus a non-polar covalent bond?
Polar has equal sharing of electrons.
Right. And non-polar is unequal. Why is it unequal? What makes it, what causes it to there to be an unequal sharing? What also is present in those bonds? Justin, now you're walking away. He answers the question and then he's like, okay, I don't know.
I'm here. I'm here.
I'm messing with you. Why, what makes it, what allows it to be a what allows it to be a polar bond? What's present there that isn't present in a non-polar bond? I'll go back to it. So polar molecules are, like Justin said, are where the electrons are being unequally shared. What's present in these three different types of molecules that's not present here? You can only get a polar bond if you have an electronegative atom, like oxygen or chlorine. Remember the six that are up here: oxygen or chlorine or nitrogen or sulfur or phosphate, that's going to have a greater pull on the electrons than the other atoms within that molecule. The whole reason that this isn't a polar covalent bond is because it's just two oxygen and they're equally electronegative because they're the same atom. So almost all of our functional groups, because we have to get it to react, and polar bonds don't react, non-polar bonds don't react with things. All of our carbon skeletons here, all these are non-polar. Because all of the electrons are being equally shared. If you want reactions to occur, you have to have non-polar bonds. You have to have either charge or non-polar bonds. And so almost all of our functional groups, actually all of the functional groups, except for this one right here, contain at least one electronegative atom. So hydroxyl is an oxygen and a hydrogen. Carboxyl, which is also a carboxylic acid, is oxygen, carbon, oxygen, hydrogen. An amino group is two hydrogens and a nitrogen. And a phosphate group, which is the kingpin of all electronegativity, has a phosphate, four oxygen and two hydrogen, making it highly electronegative. So when you add these functional groups to carbon skeletons, that's what's going to dictate if they're going to react and how they're going to react. So this is a summary, and I'm gonna zoom out just a little bit. This is a summary of all of the functional groups. I would get familiar with them. Get familiar with their names and their structures. The R, let me explain what the R is. So on each of these, R, OH, this is a hydroxyl group. Here are CH3, which is a methyl group. 2 R groups here. Think of R as being any random atom or set of atoms. Any random set of atoms. That are bound to this functional group.
Professor Gessel,
Yeah,
where you remember what part of the chapter this chart is in?
no, I don't. I can tell you that I probably didn't read the chapter for this because I lecture about this for so many classes that I don't even
All
think.
right, I'll look for it, but it does come from the book, right?
Give me 1 second and I'll look.
Okay, thank you.
Because, that's a very good question, and I just have never looked because I teach, depending on the semester, I teach 2 other general biology courses, and guess what we go over? These. And this chapter is almost identical to 1 or 2 of the chapters that I teach for those classes. So give me 1 second and I'll tell you. Hopefully they're in here because then I'm not going to be very impressed. Oh, go to, it's on 2.4. It's figure 2.13. This figure right here is figure 2.13. So this one only is giving the major ones that you find in life. However, you will find all these others also. I don't know why it's always showing you these, because you're going to see all of these as we talk.
Okay, thank you for checking.
Yeah, and I'm trying, I
So
got this,
what would
go
you
ahead.
call that table then? The one that's not in the book?
This one right here. Say it again, I'm sorry.
What would you call that? Like, what kind of title would you give it?
Let me tell you. I always put the website address down. If I try really hard that if I'm picking up something that's not in your book, or I haven't maybe copied it from another professor's lecture, which we do, I put down the website address where I got it. And I know I got that one from a YouTube video just looking at the title. But if you type in functional groups, let's do that first. Functional groups, and then I go to images. Let's see here. I'm trying to remember where I got that one. The great thing about internet is you can really find almost anything you want. And I'm gonna put something else in here. I'm gonna put in properties so that it just doesn't give me their shapes. I don't know how professors put lectures together before the internet, in all honesty. That's close, but that's not it. I must have gotten it a long time ago and I copied it from another lecture.
That's okay. So
But
title it functional groups of
let me do one other thing. Macromolecules or let's do biomolecules. Oh, you know what? And part of it is that I misspelled properties. Biomolecules. Because I know I got it from the internet. Let's see what this is now. I can't find the one I had. But the YouTube video is down below. Control copy.
So I've got my essay.
So this is the YouTube video where that figure came from. So if you want to look it up, feel free to do that. We will be talking more about biomolecules in one moment. But somewhere within this video is where that figure came from. Yeah, when I'm putting together a lecture, what I really do a lot of times is I start typing in keywords when I'm looking for a particular figure that the book does not have, that I think would be good in explaining or describing something. I go online and I look for something. And it came from this video somewhere. We may have to come back to that video in a second here. Let's keep going forward though. So hydroxyl groups, if they're electronegative, if they have the electronegative atom in them, like here where you have the oxygen, hydrogen, oxygen, 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 nitrogen, oxygen, phosphate, oxygen, 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 sulfur, and hydrogen. What that means is that that functional group either gives the molecule polar properties or charged properties. In the cases of carboxyl, or we also call this carboxylic acid because it acts like an acid. It's charged because it ionizes to release that hydrogen ion. Go ahead. Someone gonna say something or ask? Okay. Nitrogen amino group here acts like a base because it can take on more hydrogen ions. Phosphate, a lot of the way that enzymes and proteins are activated within the body is based on whether you add a phosphate group to them or you remove a phosphate group. And also ATP, which is the energy currency for the cell, has 3 phosphate groups attached to it. Nucleic acids, DNA and RNA have a sugar phosphate backbone. Sulfhydryl, which the only difference between this sulfhydryl and hydroxyl is that the oxygen is present in the hydroxyl, and in that same place in the sulfhydryl is a sulfur. What I really want you to learn is learn those 5 electronegative atoms or 6 electronegative atoms. And when they're present, you know that it's polar. And that's going to be key to how it's going to react. and how it's going to interact with other things. So functional groups, by adding them or removing them, you can change the properties of a molecule. In the case of testosterone versus estradiol or estrogen, a carbonyl group versus a hydroxyl group causes someone to become male and not female. And a methyl group. Can you find what I want you to get so comfortable with those functional groups that you can identify them on different structures? Can you find them here? Remember that R, if we're looking at this chart, that R is any random group. Okay. So here, right here, if you look right here, C, double bound oxygen, an OH, this is a carboxylic acid. This is a carbonyl. This is a methyl. This is a hydroxyl group. This is a methyl. This is a carboxylic acid again. What's acetylsalicylic acid? Are you familiar with that? You're probably more familiar than you realize. It's
Isn't it in a lot of like acne medication?
say it again.
Isn't it in a lot of like acne medication?
Salicylic acid is salicylic acid, but not with the acetyl group, not with this group. You're right. Salicylic acid is acetylsalicylic acid. And before I say the wrong thing, but I'm almost sure, I think this is Tylenol. But I'm going to make sure before I say the wrong thing by Googling it on my phone. But you're right, salicylic acid is in acne products. Aspirin, it's not Tylenol, it's aspirin. Lactic acid, what your muscles generate. Deoxyribonucleic acid, DNA. Aspartame. Yeah, if you like to drink diet drinks, this is your friend. A lot of times. So the R, remember that the R here is anything attached to that. So this is a methyl group that looks like it's that CH3 that's right here. And the R is all the rest of this. Here, this is the methyl group. And the R is all the rest of this with regards to the methyl group. This is carboxylic acid. So the R with regards to this being as a functional group is all the rest of this. Here's an amino group. Right there. So the R group is all of this that's bound to it. Questions? The most important matter in the body are the 4 types of biomolecules composed of chemical elements: proteins, lipids, nucleic acids, and carbohydrates. So these are our 4 biomolecules that, for all of the organic chemistry that occurs in our body, these are what are performing that chemistry of the organic molecules. We have some inorganic molecules like sodium chloride and carbon dioxide and water, but this is all the rest. So the 4 major organic molecules that comprise our body are what?
B.
So was that a B?
Yes.
Okay,
Bravo.
yes, B is in Bravo, yes. So first biomolecule that we'll talk about, carbohydrates. Think carbohydrates is short for hydrated carbon. So carbons that have a lot of hydrogens on them bound to them with oxygen. Remember, what's the structure of water? H2O. And so this is carbon with H2O attached to it, hydrated carbon. This group contains all your sugars, all your starches. And it contains carbon, hydrogen, and oxygen in the approximate 1:2:1 ratio. So 1 carbon to 2 hydrogen to 1 oxygen. You can be given any molecular formula and asked, what biomolecule is this? If it has the ratio of carbon is a 1, 2 hydrogen and 1 oxygen, you know it's a carbohydrate. Let me give you an example. Actually, here's some examples down here. Never mind that S right there. But glucose has 6 carbon to 12 hydrogen to 6 oxygen. Maltose, remove that S, that's a typo there, has a close ratio, 12 to 22 to 11, because the only thing missing there is 1 water, 1 H2O. Because we actually have to remove a water to make this bond, this covalent bond. So typically, these fuel sources provide the redox reactions in some of the major structural purposes, like in the DNA backbone, or they're used in cell signaling. Your blood types are carbohydrate chains on your red blood cells. We have monosaccharides, something to remember. Bear with me here. If something ends in saccharide, it's a carbohydrate. Can you hear my teaching assistant snoring? If it ends in OSE, it's a carbohydrate. Mono meaning one. Mono meaning one subunit or one structure, one subunit. Let's put it that way. One subunit. Di is one that's made of two subunits. Tri, all the fun ones, tri and so forth. Poly meaning more than one. So polysaccharide is multiple monosaccharides bound together. Disaccharide is two monosaccharides bound together. A monosaccharide is one subunit. So monosaccharides are simple sugars, building blocks of carbohydrates composed of 3 to 7 carbons. We can draw them in ring shapes like these of ribose and deoxyribose that are found in DNA and RNA backbones. Or we can draw them as a long chain, knowing in reality that they make circular structures, they make rings. If they're 5 carbons or more, they're going to make circular rings. A pentose, think of what a pentose is, 5 carbons, hexose 6 carbons. So pentose and a hexose carbons are the most important in the body. Some carbohydrates are isomers like glucose, galactose, and fructose. They all have the same molecular formula of 6 carbon, 12 hydrogen, and 6 oxygen. But they function very differently. If you've heard it as a diabetic, high fructose corn syrup is actually, it doesn't even matter if you're diabetic. It's highly toxic stuff because it's high in fructose and not glucose. And so it doesn't stimulate an immediate insulin response when you ingest it. And the result is that your sugar levels go up, but your insulin doesn't turn on, isn't activated. So you don't actually start absorbing the glucose and you just have high blood sugars. And it's because of the different structural difference between fructose and glucose that fructose is not going to initiate an insulin response. until it's able to be reconfigured and rearranged into glucose chemically. This is why I quit drinking them when I realized what was going on. Your Arizona drinks, you all know what I'm talking about, the Arizona drinks. If you look at them, they do not contain sugar. They contain high fructose corn syrup. And so when you're trying to drink them to bring up your blood sugars, like I'm prone to getting hypoglycemic. I can start to shake for just, within a matter of hours or half hour of not eating when I should have. And Arizona drinks will never fix that for me and correct that for me, because it can take an additional hour or so from when you first swallowed that drink and absorbed it into your system for fructose to be rearranged into glucose so that you can stimulate an insulin response. That's why they don't recommend it to individuals. They say glucose tabs if they're having a diabetic shock of low sugar levels, glucose tabs or orange juice or apple juice, or I think chocolate will also work, but it can't be something that's high in fructose. So how, I kind of already talked about this a little bit, but how might isomers impact physical and chemical characteristics? And of course, as I'm sitting here telling you how bad high fructose corn syrup is, I thought, I wonder what's in my drink. Guess what? It's high fructose corn syrup. I thought it was sugar, and obviously it's not. How might that impact the chemical physical characteristics? Remember how important structure is to function? Think of it as a puzzle piece. If the puzzle piece, based on its structure, doesn't fit where it needs to fit, it's not going to perform that function. If it can't fit and be bind to where it needs to bind, it's not going to perform the function you need it to. Just based on the fact that structure dictates function, and structure dictates chemical reactive possibilities. So disaccharides, molecular formulas that contain 2 hydrogens and 1 oxygen less than those observed in the 2 individual monosaccharides. Because in order to make this bond between 2 monosaccharides, you have to remove a water. So the molecular formulas contain 2 hydrogen atoms and 1 oxygen less, so exactly 1 water, than those observed in the 2 individual monosaccharides. Polysaccharides are polymers. One thing that we didn't mention, but we talked about mono meaning 1 subunit. We call it also a monomer or a polymer. Monomer is 1 subunit. A polymer is more than 1 subunit bound. two polymers of glucose. Starch is the storage molecule formed in plants. When digested, the individual glucose molecules are absorbed and provide a lot of fiber. Glycogen is the storage molecule formed in animals, which is stored in skeletal muscle and liver. When the blood sugar levels drop, glycogen is broken down at its multiple branching ends. Multiple branching ends, here's one branch, here's one branch. And the glucose molecules are individually released into the blood. So starch is a storage molecule in plants. Glycogen is a storage molecule of glucose in animals. Monosaccharides to polysaccharides and vice versa. So dehydration reactions, what does it mean to dehydrate something?
To like remove the water.
To remove a water. And so dehydration reactions are removing a water from two smaller, simpler molecules like monosaccharides to join them into a larger molecule like a disaccharide or a polysaccharide. You're just removing a water, taking this hydrogen and this hydroxyl group, removing it, making water, and making a covalent bond. Hydrolysis reactions. Think hydro meaning water, lysis meaning to break apart. So you're adding a water in order to break down a larger molecule. So rather than taking it out to produce the bond, taking the water out to produce the bond, you're adding it. You're adding it to this bond to break that bond.
I have a question.
So
So you might be able to answer this or not. So like for dialysis, a part of the process is to take the water out of like the cells and everything. Does hydrolysis ruin the cells or break them apart, or does it help them?
I don't think that this is going to be that type of reaction. I think that is more an osmotic reaction. Will you hold on to that until the next chapter?
Yeah, I was just curious.
Yeah, hold on to that. Well, you're bringing up a really good point. And I just want you to hold on to it until the next chapter, because this isn't what that is it's more of a dehydration reaction where you're removing the water. This is breaking apart carbon bonds, which that is not what you're referring to. Does that make sense? Kind of. Lauren, does that make sense?
Yes, it does.
Yeah, hold on to it. Hold on to that thought. Which reactions of dehydration reactions or hydrolysis reactions are anabolic and which ones are catabolic? What does it mean to be anabolic or catabolic?
Would catabolic be dehydration?
Catabolic be the dehydration. Why do you think that's the case?
Because it breaks, like catabolic breaks things down.
And is the dehydration reaction breaking something down or is it building it up?
No, it's building, it's bringing something together. So I don't know.
So it's the anabolic reaction.
Okay.
And therefore, Tara, what is the catabolic reaction?
Hydrolysis.
Correct. So which of these reactions tend to release energy and which ones store it?
Does dehydration release?
Dehydration release. Why do you think that's the case?
Because aren't you like taking out the water?
Yeah, I mean that would be make some sense. But let me put it another way. When you break bonds, does that release energy or store it if you break a bond?
Release?
When you break it, right. So when you store it, you're making a bond. And we're referring specifically to this bond right here.
Okay, gotcha. I just got confused. Sorry.
No, I understand. I understand. So when you're making the bonds, it's storing the energy. When you're breaking the bonds, it's releasing it. So all catabolic reactions are releasing energy. All anabolic reactions are storing it because catabolic are breaking bonds, anabolic are building bonds and storing energy in the form of bonds. What is the major function of carbohydrates in the body?
Energy.
Energy. So what number is it? You're right, Samantha. What number, what letter is it here?
A.
Energy?
Wouldn't that be
Oh,
B? Cellular?
A. Protein synthesis.
It would be what you'd use to make protein synthesis, but by itself, what is it?
B.
It's B. So because energy is fuel, right? Samantha, does that make sense? I know.
Yeah, I
Okay.
think I was getting a little confused with something else.
Well, and I think you were thinking a step ahead, and I understand that completely, because that's what we'd use it for next, right? Or we talked about energy coupling and coupling one reaction to another. So if we're doing, yeah,
Doesn't it form like ATP too? Or helps with it?
yes, yes, Samantha, now you're really jumping ahead. Yes, yes. Because what you do is you break down glucose in order to make ATP. And glucose is one of the carbohydrates. Which type of reaction occurs when biological molecules are broken down?
A, hydrolysis.
A, hydrolysis. So the next type of, any questions so far? Next biomolecule are lipids. Lipids are non-polar. So like carbohydrates, lipids contain carbon, hydrogen, and oxygen, but the ratio of oxygen is a lot less than that of the carbon and the hydrogen. There's 4 basic types of lipids. There's triglycerides or neutral fats, phospholipids. If you think about what a phospholipid, the functional group of phosphate group. So a phospholipid is a lipid attached to a phosphate group. Steroids and eicosanoids. Triglycerides are what we commonly know as fats when they're solid, or oils when they're liquid. They're composed of fatty acids, 3 fatty acids, and 1 glycerol molecule. Three fatty acids and 1 glycerol molecule. So here's our glycerol molecule. Here's our 3 fatty acids. If you think this functional group right here is your carboxylic acid. So that is what makes a fatty acid an acid. This is the fatty carbon hydrogen chain, but this is what makes it an acid. So a glycerol molecule is actually a triose. It's a sugar. It's a hydrated sugar. And it's triose because it contains 3 carbons, 1, 2, 3. And 3 hydroxyl groups, 1, 2, 3. To connect each fatty acid chain to one of the carbons of the glycerol, we remove a water. And remember what this is. This is a dehydration reaction. And we remove a water for each time, and we were able to connect all 3 fatty acids to 1 glycerol, 3 carbon glycerol molecule. If we reintroduce water, we can push the reaction in the other way. So we can break off each of these fatty acids from the glycerol molecule. What this is showing us is we talked about how dehydration hydrolysis reactions were important in making monosaccharides into polysaccharides. These reactions are the same for all of the biomolecules. If you want to build it up, it's a dehydration reaction. If you want to break a biomolecule down, it's a hydrolysis reaction. Even though we have 4 classes of complex biological molecules, the way you build them up and the way you break them down is exactly the same reaction. So triglycerides are the most efficient and compact form of stored energy. When oxidized, they provide a lot of energy. There's deposits found under the skin, aka subcutaneous, where they insulate deeper body tissues. They can vary in their chain lengths and their level of saturation. Saturation is how, for each carbon in the fatty acid chain, which is this right here, every kink here, is 1 carbon. It's not drawn in here, but there's a carbon here. There's a carbon here. there's a carbon here, there's a carbon here, there's a carbon here, and so forth, all the way down. And wherever there isn't, bear with me, I'm gonna do something different here. So here I have this structure. Like this. And that's the way it's the quickest way to draw a biomolecule when you know that it's just a bunch of carbons. Because for every kink, there's a carbon. And this end molecule has a carbon. And how many bonds can carbon take up? How many bonds can it make max?
4.
4.
4. So each of these carbons has 1, 2 bonds that are made carbon-carbon bonds, right? Carbons to carbons. Well, what are filling up the other spots so that this molecule is a stable molecule? They're hydrogens. They're just not drawn in. But chemists have just decided that when we draw a kinky thing like this one, we know it's carbons, and wherever there isn't a carbon-to-carbon bond, there's hydrogens filling up those bonding spaces, unless it's drawn in there, what it is specifically. With my kindergarten Zoom whiteboard showing. Okay. Now, what does it mean to be saturated versus supersaturated? That means that for each fatty acid chain, like this one right here, there's only carbon-to-carbon single bonds, and all of the other places are saturated with hydrogen. That's what it means to be a saturated fatty acid. It means that all of the fatty acid chain is single carbon-to-carbon bonds, with all of the empty spaces being filled with hydrogens. So saturated means like this, where they're all of the carbons are single to single bond bound. If there's a double bond, that's an unsaturated fatty acid, because it's not because there's a double bond and not 2 hydrogens attached here instead. So this molecule itself, I'll go back to it. If you're writing that down, I'll go back to it. Give me a second here. When it's like this, carbon, 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 and this. remember how many bonds each carbon can have. It can only have 4, right? So 1, 2, 3, and we can allow for 1 hydrogen here. And 1 hydrogen here. But we don't have 2 hydrogen bound to each carbon. That's why we know that it's unsaturated. Think of saturated as the level of hydrogen bonding. Or not hydrogen bonding, but how many hydrogen are bound. Questions?
Yeah, I have one.
Go.
So when they say like unsaturated fat, like is being bad for you in like oils and peanut butter, stuff like that. Why is that? Is that because your body can't process this chain or?
Are they actually bad for you? Mom? What? Are unsaturated fats actually bad for you? They're considered the bad fats. What makes them bad? She says she doesn't know. Their breakdown? Okay, they're more involved in fatty acid deposits.
Okay. Thank you.
It might be one thing that we'll talk about right now. And I wish I had an organic chemistry kit around here. I need to get one. It's one of those things I need to purchase this semester. It would be really helpful in these online classes. Is this kink here causes this whole molecule around single bonds. Each of this carbon can rotate, this carbon tail can rotate, it can kind of spin on its bond with this. This part of the molecule can spin. This right here can spin when it's a single bond. When it's a double bond, it's fixed. It can't spin. I may have to do some online shopping tonight because I really do want to. I had one for years and then I moved from Idaho and I got rid of it. And I don't know why in the world I ever got rid of that thing. Because I found it at a thrift shop and it was awesome. And they're not cheap little buggers. So I may just have to get one, even though it's going to cost me more than what I paid for that one. But what it means is that it's limited in its structure. It's not as, these are much more flexible in their structure. This is fixed. It's fixed in a plane. It cannot spin. That might be why they're found in more deposits. Is because you can't, because their structure makes them harder to break down. This is just me theorizing. It doesn't mean I'm right.
It's a good theory.
But omega-3 fatty acids, which contain lots and lots of double bonds, are actually really good for you. So go figure. But monosaturated means that there's one double bond. Polyunsaturated meaning there's more than one double bond. And what makes omega-3 fatty acids better for you than all the other unsaturated fatty acids. I don't know. Zena, I never claimed to be a nutrition specialist.
I'm sorry. I was just curious.
As I'm busy telling you the bad things of high fructose corn syrup, and I'm drinking a Mountain Dew that's the second ingredient on it. Trans fatty acids versus cis fatty acids. Trans, meaning that along this double bond, the functional groups or the rest of the molecule is going on in two opposite planes. I'm going to do this, show this differently. So this right here, this molecule is fixed. This cannot rotate. These cannot rotate. Nothing can rotate around this plane. It's stuck. It's fixed. What that means is that right through here is one side of the plane, and on the other side is the other side of the plane. This is a cis double bond. Because these two functional groups, or these two things that are attached to the carbon, are on the same plane, and the two hydrogens are on the same plane, same side of the plane. Here's one plane, here's the other plane. Cis meaning the same. If I switch this around a little bit, that wasn't what I meant to do. Okay, we're just going to get rid of it. Did you hear her really snore there? That was funny. Yes, she did sound like she was sawing logs.
I thought she farted.
No, she didn't fart. You know what? She's already done that. I just didn't bring that one up. See, she keeps me alert sometimes. It's like, oh, what was that? Okay. So here's the plane again, one side, the other side, and this is fixed. The hydrogens, the two hydrogens on each of the bonds are on opposite sides now. The two carbons that would go on are on opposite sides. This is a trans. Trans double bond. Questions? So trans fatty acids are when there's two covalent single bonds that are angling in opposites, angle and opposites on each other. They're opposite of one another. This is definitely a typo. Two covalent single bonds that are opposite of one another along the plane of that or the each side of the carbon-carbon double bond. And this is why, oh, well, no, never mind, it doesn't work. These resist enzymatic breakdown in the human body. Now, what I'm curious about is these are cis. Well, these are cis. These are cis. Okay, never mind. I think it's not that saturated fats, unsaturated fats are all bad for you. I think it's that trans-saturated fats are bad for you. And the reason is because they're the ones that remain in circulation longer, like my mom just said, because they're less likely to be broken down, thus depositing in the arteries. Does that make sense? And
Yes.
the cis fatty acids, which are all of these in the omega-3s, are not bad for you. So this is when two covalent single bonds across that double bond are in the same direction on the same side of the plane of the double bond. So are cis and trans fatty acids saturated or unsaturated?
Unsaturated.
They're unsaturated, correct. Questions? More questions that I may or may not be able to answer? Biomolecules, lipids, phospholipids. What is it? Here's your glycerol molecule. It's got two fatty acids attached to it. What makes this a diglyceride and not a triglyceride? Why would we call it a diglyceride and not a triglyceride? What's missing? Oh, you know what? Elizabeth just sent me something about good versus bad fatty acids. Well, I'll read it later. What makes this a diglyceride and not a triglyceride? Look at what's different about this in comparison to this. What makes this different from this?
That only has two chains.
It only has two chains, that's right. So this is a diglyceride, meaning it has two fatty acid chains. And on the last carbon, rather than having another fatty acid chain, it's got a phosphate group, resulting in an amphipathic chemical property. What does it mean to be amphipathic? or amphiphilic is another word for it. Someone look it up for me. Google what's amphipathic mean.
Having both hydrophilic and hydrophobic parts.
Right. Here's your hydrophobic part. Here's your electronegative phosphate with all its oxygen that are also electronegative, making it very hydrophilic because it's very polar. This is nonpolar, this is polar. Phospholipids are key to biology and physiology because they're the chief component of cell membranes. Steroids, very important signaling molecules. Fat-soluble flat molecules that are made of 4 interlocking rings. The cholesterol is the precursor for all steroids. Testosterone, estrogen, estradiol, cortisol, all steroids that are vital for homeostasis are steroids. Cholesterol is also found in the phospholipid cell membrane, where it regulates the fluidity of the membrane. So what does the public refer to when they talk about good versus bad cholesterol? They're actually referring to the lipoproteins, not the cholesterol per se. The lipoproteins are complexes that store cholesterol. So lipoproteins are complexes of cholesterol, fat, phospholipids, and proteins. LDLs, low density lipoproteins, where there's a high ratio of lipid to the amount of protein in the complex, contributes to cardiovascular disease because it accumulates along the vessel wall, forming atherosclerotic plaques, ultimately. HDLs or the healthy lipoproteins are a low ratio of lipid to protein that can may help prevent cardiovascular disease. The high density lipoproteins regulate the low density lipoprotein storage and how many are actually circulating, and promote the excretion of lipids and cholesterol. But as we're learning, cholesterol is not all bad when you need it for steroid production. You need it for cell membranes. You need a certain amount all the time. Questions? Let's take 6 minutes and get up and move around. It's been over an hour. And we'll come back to right here. I am going to try to finish this chapter. We'll see what happens, but let's take 5 minutes anyway. What are you doing? He's still sleeping.
Please leave your message. Please leave your message for
He's become a night owl, Mom. I'm gonna call Debbie one more time and then I'm gonna stop pastoring her. What is all this? Oh, it's switched, Mom. Mom. It,
Hi, Mary. Hey,
hi, it's switched.
what's next?
So I tried this monitor. The last 24 hours have not been my time for computers. I've had, they've kind of been the bane of my existence. But I could not get anything to show up on the new monitor. And it just
On the new monitor or on the monitor? I don't-
on the new monitor that I just
Okay.
plugged in. And it now is back to the way it was, but what changed? Because I didn't do anything.
So that's the way it was. What does that
Where I
mean?
can actually see things on the screen. See the files that are on the computer. And I
So is it plugged into your laptop? I'm unclear. I have no idea. Catch me up. Catch me up.
I've got to be quick. I'm only in a minute break from my class, which is still going on.
Okay.
But when I was on it earlier, it was almost like it had two screens on.
Sweetie, catch me up.
I'm
Is it
trying
on
to-
your laptop
It's
or is it-
it's on the desktop. So the monitor that you gave me,
Uh-huh.
I connected it up last night.
Okay, you hooked it up to your laptop.
No, I hooked it up to the desktop.
Oh, all right. I didn't realize that. Okay.
And then when I was trying to use it earlier, I couldn't open. It was acting like there were actually two monitors attached there when there was only one. And so there were no files showing up. I couldn't open up or view anything. And now that I've let it wait for 2 hours, it's now,
Okay, there's an option called detect. You right click on the monitor that you can see, go to, I think it's properties is where it is, and it will detect, if possible, both monitors, and it'll show the right one and it'll show the left one.
is it under display settings, Dad?
No, ma'am.
Okay,
It
it's
is
not.
right
Where's-
click properties.
I don't see a properties, that's my problem. View, sort by, refresh, shared
So
folder,
did you right
synchronize.
click on the monitor?
Yeah.
Okay,
Yeah.
then it may be settings. I thought it was properties. Go ahead.
Okay, just like, oh, why are you showing this? I don't want this. I don't even know what it is. Oh, stop, rock seal. I don't even know what it is. Go away. It's not gonna go away. So you think it's under display setting? Oh, okay. You think it's under display settings?
Yes.
Okay. All right. So rearrange displays. It has one enlarged screen. It looks like it's set up for two screens. It says rearrange your displays, one predominantly, and then there's number two. Is there any way to just get rid of the number two?
Why do you want to get rid of it?
because I want to be able to see everything I need to be able to see on one monitor. Because I only have one monitor attached.
So are the two monitors plugged in though?
I know there aren't, and that's why I don't understand what was going on earlier.
Well, that usually doesn't default back that way. Can you right click on the one, the number 2 and delete?
No, I can't.
Can you left click on the number 1?
It doesn't do anything. It just makes it, turns it colors from gray to blue.
Here's my question, is it working right now?
It is, but whether it'll be working in an hour, I don't know.
Okay, all right.
That's my issue, is I don't know what's multiple extend these displays. That might be it, because I know I've seen this before. Show only on one.
Yeah.
You know what? I'd been able to get to this earlier. I could have done this earlier because this is what I have to do at Stevenson too.
Okay.
And we like that when we want it, but we don't, when you don't want it, it's just a nightmare, as I'm learning. Okay, Dad, I've got to get off the phone, but I love you. I'll talk to you soon. Okay, bye-bye. See that earlier, I would have fixed that now. I apologize. So my dad has decided, since he's retired, to become a night person, and he's my computer guru. And he's the one who gave me this messed up monitor that I decided to try to plug in last night to help me with something. And it just made my night a lot longer. And I was saying a bunch of colorful things last night, but we won't say anything. But now I may have fixed it now that I can actually see something. So let's come back to this, and I apologize. Eicosanoids, diverse lipids that are derived from a 20 carbon fatty acid. So meaning this is 20 carbons. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and then 5 more. So these are the eicosanoids are derived from 20 carbon fatty acids. A specific 20 carbon fatty acid, meaning arachidonic acid, which is found in all cell membranes. Prostaglandin is a signaling hormone. These are like hormones, or similar to steroids in a lot of ways, but they're not steroids, they're signaling molecules. Prostaglandin specifically is involved in inflammation and blood clotting. Oxytocin, I think, if I remember right, oxytocin is a prostaglandin. Well, are we sure that, Mom, are we sure that all the prostaglandins are not hormones? She's going to get me confused in the background here. Just, you can chime in, because it won't be the first time that I was wrong. That's why I'm looking it up. Prostaglandins. Any group of compounds with varying hormone-like effects. And I'm going to look up something really quickly. Bear with me here. Fun thing of having an online class where we have Google all the time. Prostaglandin. And labor. Prostaglandins are produced naturally by the body during the process of labor. They are there to prepare the cervix and help open it in response to contractions. You're right, Mother. It's not the same as oxytocin. I was wrong. Oxytocin is definitely a hormone. So they are hormone-like molecules. They have a lot. One of the things, because as Zena can tell you, we just went through blood clotting in 122. Prostaglandins are really important in preventing blood clotting when you don't want it. Blood clotting is really great, and it's really important for maintaining homeostasis, and preventing someone from bleeding to death. However, when it's not controlled, such as when prostaglandins aren't around to localize where it's going to occur and when it's going to occur, the result is that it can be abnormal and cause major problems. Where's my screen? Of course, it took me to the very beginning, and I don't know why. Oh, shoot. And of course it died. Really? Mom, can I borrow your computer really quick? I don't think it's going to make a difference. Yeah, actually, it will make a difference. You guys remember what happened before with this? When all of a sudden we can't open the PowerPoints? Thank you, Mom. Just have enough charge. Don't, Abby, don't swat at her. Don't swat at her, Mom. I'll grab her. I don't want her to get hurt by Abby. Because we know. Keep the. As I'm sorry, I do not have Office 365 on this laptop. So when something happens to that PowerPoint, like it, why is it back there right now? This is, I told you, computers are my bane for today. Let's see here. It's still not there. What's going on? Can we move it down? But it's really, really small. Guys, give me one moment. Mouse. Where's, oh. 0903. I don't want this, I just want that. Or Mom? Mom? Oh, she went outside. Can you guys actually see that small screen? Was that a yes or no?
Which one? The
It's
one-
small, but yeah.
Yeah.
It's really small. All right, I can't. My mom's mouse, she has her computer set up so it's the opposite of every other mouse out there. Left is right, right is left. And makes me a little crazy. But let me open.
Can you hit control and then the plus sign? That should help you zoom
The
in.
control and the plus.
Yeah.
Well, actually, will that work? All right, let's do that because I cannot. I actually used her computer last night because mine was giving me such grief, but I can't make it work all of a sudden. I love computers, but I hate them at the same time. It's a love-hate relationship. Let me shut down hers. Okay. Okay. So eicosanoids made from 20 carbon fatty acids, arachidonic acid. Most important in prostaglandins, which have hormone-like properties. 3 major subclasses of lipid molecules include phospholipids, steroids, and what? I couldn't figure out how to get onto Chrome on your computer, so I gave up with Chrome. I don't know. The computers are not my friend tonight. Do you want me to get it on Chrome? No, we got this working. I'll take care of it.
Is it C?
So what's the answer? It's C, triglycerides. So we've talked about carbohydrates, we talked about lipids. Now we're talking about proteins. Proteins make up about 10 to 30% of the cell mass. They are the basic structural materials that can also be enzymes. So biological catalysts. They all contain carbon, oxygen, hydrogen, and nitrogen, and can also contain sulfur and phosphorus. The full set of proteins made within the body is defined as the proteome. Kind of like the genome is your full genetic DNA set. Your proteome is your full amount of proteins that the body can make, full set. amino acids work like a 20-letter alphabet with linkages between the letters. And the linkages that make up the letters are the peptide bonds. Or let's put it this way. The individual letters of the amino acids, the bonds between them are peptide bonds, or the linkages between the amino acids. It's the same way as it was before. Hydrolysis reaction breaks up the bond between two amino acids. Dehydration reaction creates that bond. Different amino acids have different properties just because of the functional groups. It's always going to come back to those functional groups. Most proteins are macromolecules with polypeptides containing 100 to 10,000 amino acids. However, one change in the amino acid sequence can result in a different or non-functional protein. For example, sickle cell anemia is one amino acid difference. So this is the amino acid sequence of an enzyme. Each one of the PHE, ALA is shorthand for each amino acid. So each one of them is a letter in a sentence, and that sentence is the polypeptide. If I remove a water, I make a bond between a peptide bond between the two amino acids. If I add a water, I break up that bond.
So is this an example of just one polypeptide?
Say it one more time.
Is this an example of just one polypeptide bond, or?
I'm sorry, one more time.
Just like one, like what acid sequence?
Yeah, this is an example of one amino acid sequence that makes up one polypeptide.
Okay, thank you.
So think of peptide, polypeptide. Remember, each bond between the amino acids is called a peptide bond. So a polypeptide is more than one peptide bond, or one or more peptide bond. We often use the word proteins to describe a polypeptide, but you can have more than one polypeptide within one protein. So the peptides are the links between each amino acid, peptide bonds. Polypeptides is a string of amino acids connected by peptide bonds. Polypeptide bonds, depending on the protein, one polypeptide can make up one protein. Or for another protein, there may be more than one polypeptide. For many proteins, there's more than one polypeptide. So each different amino acid is like a letter, and their various combinations give rise to the endless diversity of words and sentences. We have essential amino acids that have to be obtained in the diet. Methionine valine, threonine, phenylalanine, leucine, isoleucine, tryptophan, or lysine are all, excuse me, bear with me here. The ones that are overlapped here, actually, these are all essential amino acids. I'm just overthinking this. These are all essential amino acids. You can get these ones that are in the orange color here from corn and other grains. You can get the ones that are more in the pale pink from beans and other legumes. Non-essential amino acids are all the others that can be made within the cell. Complete proteins contain all the essential amino acids. All foods that are complete proteins contain all of the essential amino acids that you need, like milk, eggs, fish, chicken, beer, and tofu. I knew there was a benefit to beer. Looking for it eventually. I don't drink, but it sounded funny. The only time I've ever tried a beer, it was really nasty. But incomplete proteins are lacking one or more essential amino acid. So vegetables, fruits, and grains.
I think I remember hearing that that's the reason why you're supposed to eat beans and rice together, because then you get all of the.
You know what? And my mother would 100% agree with you, and she just went to bed because that is her favorite thing to eat, and she's always telling me how it's a complete protein. She likes black beans and rice, and I can only do that for so long, and then I get bored of it. And corn. She mixes corn with it. Yeah, you're right. You're right. I will let her know that you mentioned that tomorrow, because she'll be excited, because she eats it probably almost 3 times a week. Mom, did you hear her? That's why you came out. I said my mom will be so excited you said that. Legumes and rice, why do you eat them together? She said because they're complete protein. Whole cultures have been built on that.
That's a staple.
And she actually lived in Brazil for a while, and that's a staple in Brazil. Structural levels of proteins. There's 4 different levels to protein structure. The first one is the amino acid sequence itself. This is the primary structure. So looking at this would be the primary structure. How one amino acid is connected to the next. So the sequence of amino acids determined that are joined together by peptide bonds is the primary structure. The secondary structure is alpha or helix sheet, alpha helices or beta sheets being formed by hydrogen bonding between the different carbonyl and amino groups of an amino acid. So specifically hydrogen bondings using the structures involved in the peptide bonds that connect each amino acid. The third level is hydrogen and ionic bonds or van der Waals forces, and disulfide meaning two sulfurs, covalent interactions between R groups. We didn't talk about what R groups were. I'm going to back up just for a second. This structure, and I'm going right here. Here's the carboxylic acid. Here's the amino group. On every amino acid, there's one amino group and one carboxylic acid. It's where the name comes from. And then there's a carbon in between them, and attached to that carbon is on the R group, that random group that we saw for all the other functional groups when we were looking at functional groups. It's the same thing here. It's just another group. So the tertiary structure of proteins is where interactions occur between the chemical structures of one R group on an amino acid and that on another R group on another amino acid. So chemical reactions occurring between the R groups. These tertiary structures result in compact globular structures, where two are, and last one, quaternary structure. Two or more polypeptides are aggregating together through interactions. So starting at the first one, the primary level, is the amino acid sequence by itself, determined from the DNA sequence. That's what it is. Secondary structure is hydrogen bonds between the carbonyl and amino groups that make up the peptide bonds in between individual amino acids. So when one peptide bond that's connecting two amino acids is interacting with another peptide bond in a hydrogen bond. You are not going, we go through this so that you understand later, basic, when we start using this language sometimes, what it means and that it actually has a meaning. But we're kind of skating over the surface of a lot of chemistry here. Don't try to understand it in depth, because you're not going to be asked to understand it in a lot of depth. Understand the definitions.
This is just a basic overview of the
Yeah.
chemistry, right?
Well, yeah, it's a basic overview of what I would cover in 2 chapters in a general biology course. Actually, 2 chapters, almost 3 chapters. But you need it for the basis of anatomy and physiology. And so hydrogen bonds, secondary structure, hydrogen bonds between peptide bonds, the groups making up the peptide bond. Tertiary structure is hydrogen and ionic bonds, van der Waals forces, and disulfide covalent interactions between the R groups. That's the difference here. One is hydrogen bonds between the peptide bonds, between the amino acids. Tertiary structure is interactions between the R groups of the different amino acids. Another way to think of this, one other way, and I'll quit trying to elaborate on this. One way to look at this is hydrogen bonding between the links between the amino acids. And this is all the interactions between the R groups of the amino acids, not the links. Quaternary structure is when 2 or more polypeptides are aggregating. So if you remember, I said that proteins can be 1 polypeptide or they can be more than 1 polypeptide. When you get quaternary structure, you have more than 1 polypeptide in a protein. So the structure and chemical properties of each amino acid in the primary structure, remember that's just a sequence, is going to impact the overall structure and function of the final protein. Based on each R groups and each amino acid's chemistry, based on its R group, how is it going to chemically react with other things? And how could it possibly chemically interact with other R groups within that proteins, with the other amino acids within that protein? So we have 2 basic types of proteins. We have fibrous proteins and we have globular proteins. Fibrous proteins are extended strand-like structural proteins that display secondary folding but do not show tertiary or quaternary folding characteristics. Therefore, they just show the alpha and beta sheets, showing the hydrogen bonding between the atoms involved in the peptide bonds. They're very stable and insoluble in water. And they provide mechanical support and tensile strength. Globular proteins are compact spherical functional proteins. They display tertiary folding, so interactions between the R groups of the amino acids, and some exhibit interaction between polypeptides, so quaternary structure. They're chemically active and water soluble. And they play critical roles in all biological processes. Conjugated proteins can contain non-amino acid moieties, so different organic molecules that aren't amino acids can be contained within proteins. And these non-amino acid organic structures are called prosthetic groups. For example, hemoglobin contains 4 iron complex containing heme moieties. And this hemoglobin is an example of where we get quaternary structure because we have 4 polypeptides interacting together. Each of these chains is 1 polypeptide. And within each of these chains is a heme moiety, which is a prosthetic group. Something that's really important, bear with me, any questions first? I'm kind of moving along fairly fast here, but do you have questions for me? Protein denaturation. Changes in pH, temperature, and salt concentrations can disrupt the structure and thus the function of proteins. Let me give you an example. How many of you like eggs? How many of you have probably eaten a fried egg? How many, is there someone who's eaten a fried egg within the last week?
Me.
Okay, I'll let you be the one to eat all the eggs because I don't like them. But I have a question for you, Kara. How often have you been able to, you crack it, you put it in the pan and it starts to sizzle? How often have you been able to put it back in the shell and have it hatch into a chick?
0 times.
Why? It's still the same protein. It's still the same components that were there before, right? Kara,
Yeah.
I'm waiting on an answer. Why?
Oh, I don't because it's not in the egg anymore. I don't know.
The heat has denatured all the proteins, and what happens is it makes those proteins non-functional. I think
was
that
my
that's
next guess.
by far the best example that I know of protein denaturation is when you've disrupted the structure so badly that the protein some proteins can refold, but most proteins can't. And so you disrupted the secondary, tertiary, and quaternary structure to the point of where that protein can no longer perform its function. It might be reversible. Some proteins are capable of spontaneously folding back up. Most are not. Globular proteins, so these are these ones right here. Globular proteins are very sensitive to denaturation. Fibrous proteins are not. But they don't have but fibrous proteins don't have the complex folding that we see in globular proteins. Because the higher level we go up in levels of structure, the more complex they become in their folding, right? We go from the simple amino acid sequence to hydrogen bonding between the peptide bonds to interactions between the R groups and then whole polypeptide chains interacting. So globular proteins are sensitive to denaturation. Specific arrangements within atoms where active sites are disrupted. Active sites are where other proteins interact or substrates interact with that protein. And so in the functional enzyme protein structure, you have an active site exposed that a substrate binds to and you get a reaction. Well, if you shake it up and denature that active site, now the substrate can't bind because the active site is not intact. So these sites, these active sites are needed to fit and interact chemically with substrates that have a complementary shape and charge. And when you disrupt the active sites, you disrupt the functions. And if you change the structure, you're going to change the active site, most likely. Questions? This is why one of the major deficits that we have in our society, or one of the major genetics series of defects, are enzyme mutation defects. Most likely it's because you've disrupted the structure of the enzyme. And when you've done that, you've disrupted the structure of the active site. So a lot of gene therapy is to try to replace that gene with the correct gene and hopefully allow that person to have a functional protein again. But that doesn't work so well with a fried egg. Molecular chaperones or chaperonins, globular proteins that help proteins achieve their 3D structure. So these help proteins in their folding. The amino acid sequence is going to determine the precise folding and how folding can possibly happen. But chaperones are going to ensure that it's quick and that it's accurate and that the specific R group actually interacts with the right R group of 2 amino acids. And it doesn't interact with the wrong R group, resulting in a malfolded protein, misfolded protein. And some proteins will fold by themselves without chaperones or chaperones. But will they do it fast enough for life to exist, or are they going to be so slow that it's not going to happen within a feasible timetable? That's another time that chaperones help. So the major functions of chaperones, they prevent accidental, premature, incorrect folding and premature association with other polypeptides. They aid in correct folding and associations by preventing ones that shouldn't be occurring. And they promote the breakdown of damaged or denatured proteins. A lot of our diseases are the result of denatured protein plaques. Many of the diseases that you think of, especially in older age. They also will assist with the moving of proteins and certain metal ions across the cell membrane. So chaperones, the reason they were first identified, and I don't know if you can see that HSP at the bottom there. The reason they were first identified was because if you heat shock a bacteria by just putting it in a strong heat just for a minute, you'd see that the level of expression of these genes would just escalate. Because in response to a traumatic stimuli, like getting put into a desert all of a sudden, these genes would be expressed to help correct for any damages and help fix proteins and replace proteins. So heat shock proteins. So, like I said before, protein folding is crucial. We see it in a lot of diseases. How important is it? Sickle cell anemia. One gene, one amino acid defect that results in a major range of symptoms. ALS, Lou Gehrig's disease. Alzheimer's disease. Parkinson's disease. Another thing that we don't see here that Muhammad Ali struggled with was CTE, chronic traumatic encephalitis.
Isn't that where you get that from multiple head injuries?
Yes, yes, it's why, yeah, if you want to see a great movie, see the movie Concussion with Will Smith. Because that was about the man who discovered that disease. And it's rated PG-13 for a reason. There's some very graphic statements made. He talks about at one point, he says, I need to understand why a man would repeatedly pull out his own teeth and super glue them back in. Yeah, it's really, and it's a true story, and it's sad and it's harsh. And I'm not anti-football. I know I didn't watch the Super Bowl. I can't even tell you what happened in it. But he got ran out of town I think he was in I think he was in Philadelphia. And he got ran out of town by the NFL. And he is now a professor at the University of California, Davis. But the movie is based on his discovery and what he had to do to finally bring it to the forefront and make the Football League recognize and admit to the fact that it leads to chronic concussions can lead to early dementia-like symptoms.
He couldn't even go anywhere.
No, no,
Like he was not allowed to step foot on like any NFL property. Like
right,
and he was made
And-
fun of. So
right. And if you watch the end of the movie, he makes a comment. He says, I don't dislike football. He's like, my wife loves it. And he talks about how excited she gets when she watches it. He says, my wife loves it. He says, but those players need to know that this is a possibility. They need to know that they could ultimately develop dementia at the age of 45. And yeah, it's a movie. I have it because partly I like the biology in it, but it was really, really pretty disturbing and it was sad. But he's now a professor at the University of California, Davis. And it's called chronic traumatic encephalitis. And it's a it's similar to another one that we don't see here. Multiple sclerosis is another misfolded protein. And it's similar in a way, chronic encephalitis is similar in a way to MS with different types of symptoms because it's misfolded protein plaques. So a protein's primary structure is genetically determined. Any change in a protein's amino acid sequence can have various serious consequences for body cells. Oh, I'll tell you really quick, and I'm going to stop on this, not go off on this tangent for very long. I had a student last semester who was a cheerleader and struggled for weeks and weeks and weeks after falling on the asphalt with a nasty, nasty concussion, and come to find out it was her fourth one in like 2 years. And she was the flyer. I don't know if you're familiar with cheerleading. She's the one that they throw up in the air. And she ended up coming down on the asphalt on her head. And, you know, and when she told me about it, I said, you realize that cheerleading is the world's most dangerous sport. If you look it up, it's now ranked as one of the most dangerous sports out there. But she struggled for the rest of the semester because of a major concussion she got. I kept trying to get her to go and get a second opinion, but I don't know if she ever did it. Which two functional groups do all amino acids have? You guys, before you think too much about this, what was the functional group that also had an acid property to it? There was only one. What was it?
Is that B?
Say it again.
Is that B?
B is in Bravo?
Uh-huh.
Yes. So that's the carboxylic acid. That's the acid part of an amino acid. What's the other part? Don't think too much about it. Look at your choices here and tell me what's the other functional group.
F.
What?
Is it F? Hydroxyl?
No. What, look at, Zena, look at the word itself. What do you think is also there? What other functional group? You've got the carboxylic acid.
Protein, like the amino.
Yes. Guys, if you get this question on an exam, that's how you know what it is, what the answer is. Is because there's an amino group on every amino acid. There's an amino functional group and a carboxylic acid functional group. Functions of proteins do not include acting as what?
B.
B is in Bravo?
Yes.
Yep, genes, because that's DNA and RNA. Okay, so functions of enzymes, biological catalysts. They speed up biological chemical reactions in living organisms by either increasing the number of frequencies of collisions or lowering the activation energy, the amount of energy or torque necessary for two bonded structures to interact, or properly orienting those colliding molecules. So increasing the collision, increasing how well they collide, or making sure that they collide at the right angle. An idea to describe the activation energy. So this is the angle or exactness of the way that two molecules have to interact. So I made up this picture because I kept trying to describe it to students for several semesters and realized that it would be a lot better if I made a picture. So I have a reaction, and it's a reaction that I made. And in a bottle, I have a bunch of red balls and I have a bunch of yellow balls. And when a red ball collides with a yellow ball at a certain magnitude of force and at a certain angle, it will become a purple ball. So I have this bottle and I'm shaking it, and I'm shaking it, and I'm shaking it, and I'm shaking it. And 2 hours later, 2 of my red balls and 2 of my yellow balls have collided and made two purple balls. Okay. So I had, for 2 hours, I was shaking this thing, and ultimately one red ball, one yellow ball, and another red ball and another yellow ball collided at such an angle and such a magnitude of force that they reacted and made a purple ball. Well, what if I put in a structure that is capable of binding one yellow ball and one red ball and bringing them together right next to one another so that they're side by side the way they need, the angle that they need to have to react? And then I shake this ball, I shake it with this structure in there, I shake it, I shake it, I shake it, I shake it. And after 2 hours, I have, rather than just having 2 purple balls made, I have 6. So what I've really done with that enzyme is just increase the frequency at which things can react, increase the possibility of them reacting, because I've allowed something to bind to both of them and bring them to bring them into close proximity with one another at the right angle. That's activation energy. And that's how enzymes lower this activation energy. So it's really activation energy is sometimes more of a probability factor. How likely is it to occur? Enzymes make it much more likely to occur. So enzymes allow reactions to occur at a normal body temp. Enzymes are going to decrease the randomness of reactions by binding to reacting molecules temporarily, like my yellow and red balls here. And presenting them to each other in just the proper positioning for a chemical interaction. So the basic steps involved is that first the enzyme binds a substrate, which ends up making the enzyme change its structure and kind of distort the bond between the two parts of the substrate. And then the enzyme substrate complex undergoes an internal rearrangement to break the bonds there, or to make a bond. Either way, enzymes are involved in both. And then the enzyme releases the reaction product. So it just, the substrate binds the enzyme. Once it binds, the enzyme kind of shifts its structure just a little bit and kind of distorts the connection between the two structures of the substrate. And then it breaks and the enzyme releases the substrate, the products. Questions? Let's, you know what? I think we should end right here. I plan on finishing this on Wednesday night, when we have lab. So after we have lab, the integumentary system PowerPoint is already up. When we have lab and after we're done going through that PowerPoint, I want to finish this chapter. We don't have very much left. And have a wonderful night.
Thank you. Have a good night.
Thank you.
Thank you.
Thank you.
Thank you.
I just have a quick question.
Yep.
What is due for, I know what's due for lab, but what is due for lecture?
I don't
This
remember.
week.
I don't, you know what? Nothing should really be due for this week because we're going to push it back because we didn't finish this chapter. I don't think there's anything due this week for lecture.
I just want to check because I know it doesn't follow the syllabus and sometimes
because we're pushing things back as we
Yeah.
don't get it done.
Yeah. I just wanted to make sure I'm clear, to make sure I meet my deadline.
Nope, I understand. Let me see. Can you find these deadlines on the, do you see where they are on D2L?
Yes.
Jennifer? Okay.
Yes. And that is kind of what I've been following, not the syllabus. I
Right.
look at the syllabus to get an idea.
I would follow D2L more. Yes. But let me look and let me just see. So none of this is due until next week. So there's nothing due this week.
Okay.
And I'm going to push back these chapter 3 for another week.
Okay.
But
Thank you.
nothing is due this week for lecture.
Okay. I just like to write it down just. to
Yeah,
make sure.
the chapter 2 tends to take the most amount of time. And
Okay.
we're actually working through it faster than other semesters have gone by.
Okay. Sounds good. Thank you so much for clearing that up.
You're welcome, of course.
Have a great
I
night.
also have a quick question for you.
Oh, go.
Sorry.
No, don't be sorry.
I should have emailed, but I honestly I forgot
Tara,
until she just said
Tara,
that.
it's always a good idea just to ask me in class, I promise.
So I've just been like checking up on my graded assignments and everything,
Huh?
and they usually all come up pretty instantaneously,
Right.
with the exception of it's still saying it doesn't have a grade for my chapter 2 quiz, and I know I've taken it.
For the chapter 2 quiz. Can you send me a screenshot showing that you've completed it?
I don't know how to do that. Would I just go and pretend like I'm taking it
Can you-
again and it would say,
Actually, can you go into your McGraw here? Let me stop recording.